Muscle physiology, this is the first, we're doing our review. This is our Unit 2 exam review. 4 parts: muscle structure, excitation, contraction, coupling, metabolism, as well as non-skeletal muscle. For part 1, you should know the connective tissue layers surrounding muscle. Who could tell me the 3 layers? Epimysium, and what does it cover? The whole muscle is epimysium. The next one, that's around fascicles or bundles, right? Endomysium is around a single, just one muscle cell. Perfect. Connective tissues and tendons and ligaments. What's the relationship between the mysiums, the 3 mysiums we just went through, and a tendon? So what? It's like a Tootsie Roll. Excellent. So it is like the paper of the Tootsie Roll, where it's just the ends. And so you can see how it's connected through from not only each individual cell, but bundles, and then the whole muscle. So that's what makes tendons so strong, from the point of origin and insertion on a skeleton. Components of a muscle fiber. So you should know the basic anatomy of a muscle fiber. The cell membrane. So a cell membrane is sarcolemma. Inside, the cytosol for a muscle cell is called sarcoplasm. What is the organelle that contains calcium? Sarcoplasmic reticulum. What is the part of a muscle cell that conducts electrical and impulses into the core of the muscle cell? T-tubules. Fantastic. What is part of the cell that's actually doing the contracting? Myofibrils. Fantastic. And then you go down, the myofibrils are made up of what 2 types of proteins? Actin and myosin. Which one's the thick filament? Myosin. Which one's the thin filament? Actin. Which one has active binding sites? Actin has it. Which one has the cross bridges that want to bind to the active sites? Myosin has the cross bridges. So when we don't want to be contracting, we have to cover those up. So these are regulatory proteins. What is a regulatory protein that's wrapping around actin that's covering up the active sites that when we want to relax? Tropomyosin. And then what is the boss of tropomyosin that just sort of tells it it's time to uncover or it's time to cover? Troponin. When it tells it to move over, tells tropomyosin to move over or not, how does it do that? Calcium. So the fact that calcium is going to bind to it, when calcium troponin complex, when they're together, that's what's going to cause tropomyosin to expose that active site on actin, and then cross bridges can do their thing. The components and regions of a sarcomere. I really care about what are the ends of a sarcomere? Z-lines, Z-line to Z-line. Okay, excitation, contraction, coupling. The neuromuscular junction, what is that? What part of the nerve? Tell me about the neuromuscular junction. The action potential has arrived from the axon, and it's at the end of the axon, has a part of a neuron that's called what? This whole neuromuscular junction is a synapse. Exactly, it is a synapse that we went into just generic detail when we did neurophysiology. This one's specific from the end of a nerve to a muscle. So we have an action potential that came down the axon of a neuron, goes to this bulbous end of the neuron that's called the axon terminal, or there's other names, synaptic bouton. There's a space, the synaptic cleft, and then there's a sarcolemma. So the neuromuscular junction will release a neurotransmitter that will cross the cleft, and if this table here is the sarcolemma, the muscle, there'll be little receptors. So a little neurotransmitter will bind to the receptor, cause opening of some channels on the muscle cell, causing the muscle cell to become activated, or we know the word is actually depolarized. So the whole sarcolemma of a muscle cell will depolarize, and then it's going to go down the little T-tubules. They're going to get depolarized. And then the T-tubules down into the core of the muscle then tells the sarcoplasmic reticulum, let the calcium out. And then we know we can start the whole troponin, binding to troponin, and the cross bridges and stuff. So the role of nerves in activation, I wanted you to know that you have to have a nerve going to every single individual muscle fiber in order for it to contract. But you can have one neuron with lots of little branches off. So one neuron can serve many muscle fibers or muscle cells. What is that called? Motor unit. There we go. So it's a motor unit. So a single motor unit means you have one neuron with kind of lots of branches that's going to activate a certain number of muscle cells at the same time. So, the process of contraction did the whole neuromuscular junction, depolarized the muscle cell membrane, activated T tubules, and the T tubules told the sarcoplasmic reticulum, release the calcium. Calcium is now free in the cytosol. Calcium binds to where? Troponin. Troponin then moves over tropomyosin. Active sites are now exposed. What's going to bind to them? Cross bridges or lift. That's the first thing it does, and it pivots over. So it actually pulls over the actin a little bit. We're going to then need what to detach? Excellent. ATP to pull that cross bridge back down and reset it, and then it pops back up and keeps going, and we have more ATP to pull it down. And it will continue that process and continue to contract as long as there's ATP available and as long as those active sites are exposed. When the muscle goes to relax, there is this pump on the sarcoplasmic reticulum. What is that pump going to pull? Calcium. And it's going to pluck calcium off of the troponin. Tropomyosin goes back over covering those active sites. The cross bridges are like, they don't have anything, so they're just going to lay there. So now there's no cross bridges being bound because now the tropomyosin is in the way. And calcium is getting brought back into the sarcoplasmic reticulum. It takes ATP because we have even more calcium hidden in the sarcoplasmic reticulum. So we're kind of shoving it back into an already full one. So it's going against its concentration gradient. As long as we're covering up those active sites on actin and cross bridges are no longer made, that's what relaxation is. So we're sort of reducing the cross bridges and the force is going down until we're finally completely relaxed. The three phases of the twitch: the latent period, so the whole long stuff of like neuromuscular junction, acetylcholine binds the sarcolemma, depolarizes it, T tubule, sarcoplasmic reticulum, release calcium, bind to troponin, move over tropomyosin. All that happens in the latent period. Then as soon as the first little crossbridge lifts up and moves, we're now in the contraction phase. Contraction phase is just binding of cross bridges and they're pulling. Then the relaxation phase is after that sarcoplasmic reticulum calcium pump kicks in and it's sucking out all the calcium, and now we're slowly covering up more and more active sites, so we're detaching cross bridges, and then we're having less and less force until they're all detached and we're fully relaxed. The types of twitches you should know are the single twitch. So we just talked about the three phases of the single twitch: latent period, which you need to know, contraction, and relaxation. And then there were many types of multiple stimuli. And the point of the multiple stimuli is that you could rapidly stimulate a skeletal muscle to trick the sarcoplasmic reticulum into releasing more calcium, and more cross bridges ultimately are bound. So that makes more force. Instead of learning all of the smaller steps, I just want you to know a single twitch so you know the phases. And then what is maximal force? And that is going to be tetanus. Tetanus is where you're stimulated the muscle cells so much that you have as much calcium out as you can get, and you cannot get any more cross bridges bound. That is the maximum number. So that's the maximal force a muscle can make, is tetanus. The last part of this section is the types of contraction. We have the isotonic phases. Eccentric is if you have a barbell and you are slowly letting it go down, and so your muscle is elongating. That's eccentric. Concentric is bringing and your muscle is shortening. The isometric is if I were to just sit here and pull on this table, but it's bolted to the ground, my muscle will contract, and after the initial contraction, it doesn't shorten anymore. And so once it's kind of pulled up the slack as far as the connective tissues go, it will not shorten. So that's isometric. Muscle metabolism, the role and needs of the mitochondria. So the role of the mitochondria is to produce a whole lot of ATP. Because how much ATP do we produce if we just have the cytosol? Two. That's pretty pathetic. How much for a single molecule of glucose if we have mitochondria? Yep, anywhere from 32 to 36. The mitochondria is where the Krebs cycle and electron transport take place, and we get a lot more ATP. But what do we have to give the mitochondria for us to get the extra ATP? Oxygen. Fantastic. Oxygen is required in the mitochondria in order for us to get the maximal amount of ATP from a molecule of glucose, as well as ATP from proteins, and most importantly, fat, which is one of our best sources. So ATP production, you should know that it's glycolysis in the cytosol, and you only get 2. Then we move into the mitochondria, and we get more, but we have to go through the Krebs cycle and the electron transport chain within there. So as far as muscles go, the muscle fiber type are slow twitch fibers, which are known as red fibers because they contain myoglobin. What does myoglobin do? It pulls more oxygen out of the blood, so it brings extra oxygen into the muscle cell so you can better utilize your mitochondria. So it's ideal for marathon runners, long distance aerobic activity. Fast twitch fibers, they're the ones that will enlarge with hypertrophy, with strength. So the more force that you put on it, the more that it will accommodate that force by making more actin and myosin, so it can be stronger. But fast twitch are also quick, and so they respond to rapid movements. So those tend to be white fibers because they're such short duration acting muscles, they don't need that extra myoglobin. They're going to deal with an immediate energy source known as creatine phosphate. Fast twitch fibers, they are, again, very short duration bouts of intense exercise. Cardiac and smooth muscle. So we've talked in excruciating detail of skeletal muscle. So we'll just do a little light version of cardiac and smooth. That we know cardiac muscle is the heart. Smooth muscle is all of our autonomic organs. It could be our esophagus, swallowing food down, moving food through the GI tract. It's around the uterus. It's around blood vessels. It's going to dilate. It will relax to lower your blood pressure. It'll constrict in blood vessels to raise your blood pressure. But those are things that you don't have control over. So those are autonomic. They're really around dealing with our organs, our arteries, things like that. Skeletal muscle is the only one that has more than one nuclei, so considered multinucleated. Skeletal muscle is also the only one that is voluntary in that you have to initiate a thought in your brain and the nerves have to go down in order for it to activate. Smooth muscles don't need nerves to activate, nor do cardiac muscle, although they do have nerves to influence its activity, but they could actually be stimulated to contract without nerves, unlike skeletal muscle. Smooth muscle is considered smooth because it doesn't have the dark light bands that are the striations, so it's not as organized as skeletal and cardiac. The summary for the Frank-Starling law of the heart means that as you have greater amount of blood returning to the heart, say in an exercise capacity, the chamber, the ventricular chamber is going to stretch. It's going to fill up a little bit more like a water balloon, and those fibers are going to pull apart. And when they pull apart, all of a sudden the troponin actually gets more sensitive. And so even though you have the same amount of calcium coming out, more tropomyosins are going to actually move aside, and more cross bridges are going to be formed. So when you have more blood coming back to the heart responds by contracting more forcefully. So that's the main point here. The Frank-Starling law of the heart is due to cardiac stretch. It will respond by contracting more forcefully. And that's it for our muscle physiology. Neurophysiology, part 1, remember, was just the cells. What's in, what are neurons? What's in this neural tissue? We're going to talk about the resting membrane potential and the active potential. So this is the voltage changes, the actual synapse of how we have a neuron connecting with another tissue, and then specific neurotransmitters. So what are the 2 types of neural tissue? A neuron and neuroglia. Exactly. The neuroglia is a lot of supportive roles, so we don't need to name the specific neuroglia. As far as the different groups, there is one neuroglia we do need to know. Who remembers what that one is? It's the one that forms myelin. The Schwann cells. Excellent. The types of neurons we should be familiar with. There's two real main ones. We have interneurons, which are just the connectors. But what are the two ones that I care that you know about, because we care about what their shape is, because the shape of these neurons make a difference to what's going on when we get to the spinal cord part. So what are these two? A motor and sensory. Excellent. Motor and sensory neurons. A motor neuron is going to look something more like it has these guys sticking off, which will be dendrites. And it has an axon, and the axon can then branch off to various axon terminals, whether it's going to many muscle cells or it's going to parts of a gland or whatnot. So this would be more of a motor neuron. And a sensory would have dendrites that would be receiving, going also to an axon, but it has the cell body off in a little cul-de-sac here. And then it's going to go and it's going to have its own, we'll just sort of call this axon terminal, where we have neurotransmitters, just like we do in here. So that's going to be sensory, that's going to be motor. Their shapes will make a difference with regard to the spinal cord anatomy. The anatomy of a neuron, so we name the parts. You should be able to identify if you were to see a picture, and if I showed you a picture of a neuron to do the anatomy on it, I will do a motor neuron. You should know the dendrites. Cell body with a nucleus, axon, axon terminals. Motor neurons, some are myelinated and some are unmyelinated. If they were myelinated, what does it look like? And where would the myelin be?
Along the axon.
They're going to be on the axon. And then what do they look like on the axon? It's like the whole axon just covered in it? They're all hot dog buns, one after another. So there's going to be a segment that has this myelin sort of wrapped up in it, and then there's going to be a gap where you still see the axon. Then there's another section that's going to be wrapped up in myelin, and there'll be another gap. So you'll see sections of it. So myelin is formed by Schwann cells. It will be on the axon. The function of myelin is to speed the conduction down the axon. So it will make this action potential process that we're going to do next more rapid. Helps insulate neurons from its neighboring neurons. It also protects it and helps guide neurons that are damaged. It provides a path for some of these neurons to regrow out. Regrowth of a neuron requires two important things that are absent in the central nervous system. So when we regrow damaged neurons, this takes place only in the periphery. What do we need to happen first after damage occurs? Cleanup crew, exactly. Macrophages. So macrophages have to go in and eat all the broken debris, clear the area, and then we have growth factors that are going to be released, and that helps the end of the axon start to grow outward. The myelin will help guide it, sort of like a threading the needle, and help guide that axonal growth back to where it was going to go. We don't have the cleanup crew, and we have inhibitors of some of the growth factors in the central nervous system. So if we damage neurons in the central nervous system, our growth is either very, very limited or non-existent. What are the two ions that you guys need to know about that determine membrane potential? Sodium, potassium. Those are the only two that we're going to worry about. There's others that are involved, but we're just going to focus on sodium and potassium. Where do we have a lot of sodium? Outside extracellular fluid. Where is there a lot of potassium? Its highest concentration will be intracellular, inside the neuron. Perfect. So they're going to be on opposite sides of the membrane in terms of their concentration gradient. We have a little bit on both sides, but the bulk of it's going to be inside for potassium and the bulk of it's going to be outside for sodium. Channels. We have two main types of channels. We have chemically gated channels. So they would be on a membrane. The channel's got a closed lid, but it's going to have a little slot here where a neurotransmitter would have to bind to it for that channel to open. That's going to be chemical. So it's only going to be in a location where it's going to get a chemical to respond to. Those are chemically gated. So they're going to be found where? Yeah, so you're going to find these guys on dendrites. So if this is our neuron here, we've got a little, we would have these chemically gated channels located here. So we'll put these guys. So that another neuron that's coming elsewhere would have its axon ends, you know, the axon terminals that's going to go to it. So this is where we have chemically gated channels. We can have a chemically gated sodium channel or a chemically gated potassium channel. So because one channel only serves one ion, so they don't have both of them, they don't share. Voltage gated channels are the other ones. Where are they going to be located? They're going to be located on the axon. So we have little channels all along here. So they're going to be on the axon. They're going to be either potassium, which would be K+, or they're going to be sodium. Because it's voltage gated, it doesn't care about chemicals around, but it cares about the voltage. So at a specific voltage number, they will open, and at a specific voltage value, they're going to close again. Potassium will open at what voltage? Plus 30, exactly. And they're going to close at what? Minus 70. We're just going to say back when it gets to its resting. And you always want to make sure you have the millivolts down. What about sodium? Sodium's going to open at what voltage? We'll say minus 55, yes. We're around there. That is also known as threshold. They're going to close at what? Plus 30. So I want you to see, just because you have plus 30, it's actually the opening trigger for one channel, but plus 30 is the voltage that's going to shut down another channel. So they're not all going to be the same. They just really have a voltage level that will open it and a voltage level that will close it. What about a membrane pump? What do they do? So we have membrane pumps. That's going to be here, these green circles, but we also have them up here. So they're all over. A membrane pump are known as the sodium potassium ATP pump. It's going to move 3 sodium for every 2 potassium. So it's going to reestablish the gradient. So when we open a channel, sodium's going to go into the cell and potassium's going to leave. But the cell wants to get back to normal. It wants to bring its potassium back in, and it wants to kick the sodium back out. So the pump is going to reestablish that balance. What is a graded potential? You can have a graded potential that is depolarizing. So if it's depolarizing, it's going to be more positive. But it's graded so it didn't hit the threshold point. It just means, okay, we got a little bit of positive, and the pump's going to make everything go back to normal, so we're back down to resting again. Or you can have a graded hyperpolarizing one, and it made it just more negative, and then nothing really happened, and then the pump reestablished it and made things go back. Graded just means you can either depolarize or hyperpolarize, but you hadn't really activated the cell yet. So several graded potentials on a neuron can actually cause it to reach that threshold point. And then you have a full-blown action potential. So you should know about our action potential diagram. We have our millivolts is our voltage of a cell. The cell starts off at rest. What is the resting membrane potential of the cell? Minus 70 millivolts. And we know the sodium channels are going to open at minus 55 millivolts, so that's known as our threshold point. So at rest, it may get a graded potential, and maybe it gets several. So the graded potentials are summed together that we reached this threshold point. And so then all of a sudden that we shoot up, this upward movement means that sodium is entering the cell. I'm going to say enters the neuron, because at this point, our sodium voltage channels are open. So at the dot, they opened, so sodium is going to now enter the cell. We reach the peak, which is plus 30, and those sodium channels close. It's also at the peak, another channel opens. As our potassium voltage gated channels will open, and then it causes our cell to go down and become less positive and more negative. We finally shut those down about here, but we kind of go beyond it and come back up and go back to minus. So during this time, we know we have sodium. I like to draw it like pretending this is a cell. Sodium going in the cell This is known as depolarization. And then during this phase, we have potassium leaving the cell, and this is known as repolarization. And then in this phase, I'll just draw it as a circle, we have our 3 sodium going out, and we have our 2 potassium going back into the cell. So it's the pumps, and it's also, that phase is known as the overshoot, hyperpolarization, undershoot, look at a couple terms here. So it's really just rebalancing during this phase, and then we're back to normal again. So you should be prepared to see a diagram of an action potential and be able to know what's happening at each phase, what channels are open, what that phase is called, or what's happening. So that's the action potential diagram. Within here, we also want to know about refractory period. There are 2 refractory phases. Who can tell me what the refractory period is all about? There's an absolute refractory and a relative refractory. Absolute refractory means you cannot get another stimulus in this neuron. So from this point, when we close our sodium voltage gated channels, all the way down until we get back to -70 again, this phase here is our absolute refractory period, that another stimulus will not do anything to this nerve. It's just these sodium voltage gated channels are considered to be locked. They're closed and locked, and you can stimulate all you want, but they're not going to open again. The next refractory period is known as relative refractory period. And this means the cell is now negative. It is still not back to normal. So it is less likely to want to make another stimulus, but it could if it had a larger than normal impulse to make it do so. So instead of just reaching threshold of that -55, it's going to have a larger series of greater stimulus to get another thing going. And that happens at the same time as this hyperpolarization phase is occurring. And so this means it can do another impulse, but it is not as likely to. Tell me about continuous propagation and saltatory propagation. What is continuous?
It's like a domino effect.
Yes. So continuous would be if we had our neuron, our dendrite cell body, and we have this long axon, and we already talked about the sodium channels, and they're, we have sodium channels all the whole distance, and we have our potassium channels all the whole distance, and we have the pumps and all that. So we may have an action potential that happens here, causing the sodium to go in and the potassium to go out. So that would be in this section. And then all of a sudden it's going to happen to this section. So sodium goes in, potassium goes out. So it's going to happen to this. And so it's going to trigger this one and this one. It happens there. And so you have this action potential process that we went through with the graph, which is telling you about what's going on here. But it's going to trigger another one that's going on next to it, and it triggers another one. And so it does it in a very systematic, continuous manner. So again, like dominoes, one section triggers the next, which triggers the next. This is considered a little more slow. So if we want to speed this process up, what do we want to add? Exactly. So if we add myelin, we're going to have little sections here. We have like a little myelin section. That's right. So then we only have our sodium going in, potassium going out, and it sort of beams across. And then we have in and out, but it comes over here, in and out. So whatever is activating at this point will stimulate something much farther down. So this here is known as saltatory conduction, and it is much more rapid. A synapse. This is the same as we talked about a neuromuscular junction. It just happened to be a synapse at a muscle. A synapse is just like this picture where we can see the end of this first neuron is going to then release neurotransmitters onto the dendrites of a second neuron, or it could release onto a muscle. So we could have dendrite cell body axon going to another neuron, or we can have it coming off going to skeletal muscle. It just is anytime you have the end of a neuron and the start of another cell. So these areas would be where a synapse would take place. If it's a muscle, it happens to be called a neuromuscular junction, but it's still a synapse. That's just the general term. So you should know the parts of a synapse. You have the axon terminal of the first neuron. You have the cell membrane of the receiving neuron, also known as the postsynaptic neuron. There will be channels. Remember, this is where we're going to find chemically gated channels. And so when a neurotransmitter binds to it, it can open. They're usually depolarizing sodium channels, and it's going to get more positive in here. And if it gets positive enough, it'll tell this neuron to make an action potential. If it gets enough that hits the 55 for the axon. If it's a muscle, you're going to send the acetylcholine or the neurotransmitter out. It's going to bind to a receptor, open up sodium channels. It's going to depolarize and activate the muscle cell. So that's all a synapse is. We have the components. You have the cleft. Who remembers how do we, once we send a neurotransmitter out to a receptor, does it just stay on the receptor or what happens to it? There's an enzyme. So we have enzymes that then break it down, break it apart so that we can then send it back up here, repackage it, reform it, and use it again another day. There are only two types of neurotransmitters that don't do that, and those are our neuropeptides. Do you guys remember which ones those are? Endorphins and substance P. Otherwise, anything else we talk about does it that way. Neurotransmitters, this is just your list. So we talked about the synapse, and one neuron releases neurotransmitters. It could be any of these. They just do different things in different parts of the body. Questions about the neurotransmitters would be something along the lines of, which of the following neurotransmitter is involved in muscle contraction? And that's where we have neuromuscular junction. Or somebody with Alzheimer's disease is going to have issues with which neurotransmitter? So then both of those would be acetylcholine. Which three would be involved in depression? The next three on the list: norepinephrine, dopamine, and serotonin. So those things are involved in depression. We happen to know Prozac is specific to serotonin. You should know that dopamine is, again, all three of them are feel good. Dopamine is going to be with attention deficit hyperactivity disorder. I don't think I said what ADHD was, as well as in a different part of the brain, Parkinson's disease. That's associated with dopamine. Norepinephrine also is considered to be one of our feel-good neurotransmitters, but an excessive amount can lead to aggressive behavior. Glutamate is going to be, think of your learning and memory, and you don't want to drink alcohol with it because you're going to impair laying down those memories. So you want to be able to establish some long-term memories. Alcohol interferes with that. GABA is going to be our only inhibitory neurotransmitter. If someone doesn't have a sufficient amount of GABA, they're going to have more anxiety, but most importantly, epilepsy. They're going to be more prone to random things stimulating, causing action potentials. And so then you can have inadvertent activity of the brain that wasn't initiated. And so that's really what an epileptic seizure is. Substance P and endorphins, those last two are the only ones that don't recycle at the end of the neurotransmitter because they're considered to be neuropeptides. All I want you to associate with substance P is pain, and endorphins are going to be pain suppression. So they're the opposite of each other. The other thing that endorphins is important is that exercise can boost them. It has to be chronic over several weeks and has to have a level of intensity with it. But morphine or the opioid drugs actually fit into these endorphin receptors, which is part of their appeal to people that tend to get addicted and abuse these drugs, because it does make them feel good, but often it begins by pain suppression. So that's how people initially get into that. Okay, spinal cord, nerves, and reflexes. We're going to first have you know the parts of the spinal cord. You're going to need to know the nerves coming off of the spinal cord and the reflexes, the reflex loop that is included there. Who can tell me the 3 meningeal layers protecting the spinal cord? Dura, arachnoid, and the pia. Which one is touching the surface of the spinal cord? Pia. Which one has cerebral spinal fluid flowing within it or below its space? Arachnoid. So it's technically the subarachnoid space, but it's confined within the arachnoid because it's all fibrous. The fluid can make it through there. And the dura is this tough outer layer. So the spaces between the meninges, the most important one is going to be that subdural space because of the cerebral spinal fluid is going to be within there. The spinal cord regions, cervical is our neck region, thoracic is along the region of our mid-back, and we have our lumbar, and it goes down into our sacral region. So we have nerves that come off of these regions, as well as a coccygeal region. We don't want to talk about that one. But we have nerves that come off of each of these sections, and they'll be known as cervical nerves or thoracic nerves or lumbar nerves. That's where they get their terms. It's going to be wider near the cervical region, and it's wider down in the lumbar and sacral regions as well. The end of the spinal cord is where? It doesn't go all the way down to the bottom to where our butt is. Where does it end? L1 and L2 is the end of the solid mass of the spinal cord, but hanging down and continue to go down are going to be a bunch of spinal nerves. So they will be within this region. What is it called that's down there where it's just the stringy nerves? It means the horse's tail. It's the cauda equina. That's right. So then, so that's the cauda equina. That's where you're going to want to go if you're going to do a spinal tap on somebody. You're going to be cerebral spinal fluid, but you're less likely to poke the solid part of the spinal cord. And what is gray and white matter? Okay, so if we have this 2 neurons, we have this neuron and that really malformed neuron over here. Which part on here would you consider to be white matter and which would you consider to be gray matter? So we'll do white here in red. So this would be considered the white matter. And the gray matter is going to be here where these connections are taking place. We have gray matter here. So what's in gray matter would be axon terminals, dendrites, and cell bodies. And so the synapse is happening within the gray matter. The cross-sectional anatomy of the spinal cord. So if we cut across a spinal cord, it has gray matter inside, which is shaped like a butterfly, and there's a hole in the middle. So let's name the parts of the spinal cord. What is this back part here? Yep, this is going to be the posterior horn. What about this part here? Anterior horn. What about this part here in the middle? It's going kind of connecting the 2 sides. That would be the gray commissure. And then I didn't draw in. I didn't do a very good job this here. It kind of goes a little crack there. So this little part right here is anterior median fissure. And what's all out here in these spaces that's outside of the butterfly? That's going to be white matter, yeah. So who can tell me, while I'm erasing this, what nerves are and what type of neurons can be found or what's found within a nerve? So we'll draw this as a nerve is going to be way out here. It's past the confines of what the vertebral column has. is going to divide, or we have the spinal cord within here. We have, let me put this in green, a cell body here in this anterior horn. So this is, I'm drawing a cell body for what type of neuron here? And it's going to come out, and it's going to go all the way out here. We have its axon terminal, and it might go to a muscle or something. So this guy here is going to be a motor neuron, also known as efferent, because it's going out to the periphery. Then we have our sensory neurons. We have dendrites, because its receiving portion is out in the periphery, has an axon coming in. It actually has a cell body hanging out here because the sensory neurons are shaped differently. And it's going to have its little axon terminals back here in the posterior horn. So this here is going to be a sensory, also known as afferent. The two together, or the two types, it's sort of confined within here. So this here is going to be, that's going to be a spinal nerve as it first comes off the spinal cord, and then beyond that it's just considered a nerve as it continues out. The spinal nerve is just the initial projections as it comes off of the vertebral column, but they could just be called nerves as they continue out. But the point of the nerve is that they're mixed. That has the motor and sensory together. So where they're separate, what is this section called? This little guy here coming off the spinal cord is known as the ventral root. And this guy coming into the spinal cord is known as the dorsal. So the dorsal root would have axons of sensory neurons. Ventral roots would have the axons of motor neurons. Because the sensory side of things has weird cell body located out here, there's a wide area known as the dorsal root ganglia, where there is sensory neuron cell bodies. So you need the posterior horn, anterior horn. As far as the putting a neuron with it, anterior horn has motor neuron cell bodies. Posterior horn has axon terminals for sensory. The spinal nerves, so when we talk about the spinal nerves, it's already when it's out here, you know, after it's not the split, not the dorsal. We're outside the spinal cord, now we're talking about spinal nerves. So you should know the number. How many cervical spinal nerves do we have? 8. Thoracic is 12, lumbar is 5, sacral is 5, and we have 1 coccygeal one, but we'll not talk about that one. Types of neurons that make a spinal nerve. That's what I meant here. It means I meant motor and sensory, that you know that they're mixed. Connective tissue layer around the nerves, endoneurium around one, perineurium around a group of them, and epineurium around the whole entire nerve. Dermatomes, they are strips of skin that are sensory for a specific nerve. So when I said we have 8 cervical nerves, they're named as C1, C2, C3, all the way through C8. So there are specific strips of skin that you could touch, and if someone is numb at that spot, you can identify that they have a problem with a specific nerve number. So those dermatomes are a very precise body map of the sensory receptors for each of those types of spinal nerves. So for the plexus, who can tell me the 4 plexuses that you need to know? Cervical, brachial, lumbar, and sacral. So for each plexus, you need to know the name of the plexus and the spinal nerves that go to it. So what would be cervical plexus? C1 to C5, and what's the main nerve that you need to know from that plexus? Phrenic, and what's it do? Breathing, diaphragm, breathing. brachial plexus, that one's the hard one. There's 5 nerves. C5 to T1. So you need to know that. So a test question might be, the spinal root C5 to T1 is which of the following plexuses? So I have them listed. You're like, okay, C5, T1, brachial. And you'll pick that B or whatever letter it is. Or on the lab practical, I may have, hey, here's the plexuses, tell me the spinal nerves, and you fill that in. Or I give you a fill-in, say, you know, C1 to C5. Then you say it's cervical plexus. Or I'll say breathing. What's the nerve that goes to that? Phrenic nerve comes from the cervical plexus. Of the brachial plexus, you have the axillary, which is going to go to the shoulder area. You want to think of radial in general as just the back of the arm. You want to think of musculocutaneous as being controlling your biceps. And then on your forearm, you want to think of the ulnar is your pinky, and it's going down to your medial forearm, because where your funny bone would make you feel weird, to your pinky and part of your ring finger. That's your ulnar, because it's on the ulna, where the bone is. And the other one, that's going to be your median nerve. The legs, the lumbar, the main one I'm going to have you focus on is the femoral nerve going to the front of the thigh, which is your quadriceps muscles. Sacral plexus goes to the sciatic nerve, which comes off and is going to go to your genitals as well as your posterior leg, your entire back of your leg. But think of the back of the thigh is going to be your sciatic nerve. But you get to the level of the knee and it's going to split. You have the peroneal or fibular, which is going to go out on the lateral side, and the tibial, which just sort of stays down the rest of your leg. So you pretty much have the side of the bottom of your leg or the rest of the bottom of your leg as far as the tibial. So the main thing is for each one, know the spinals, comes off to the plexus and an important nerve from there, and generally what that does in a very general area that it serves. Front of thigh, back of thigh, outside of lower leg. Like, I don't care that you know about movements or anything like that. Breathing, diaphragm for the phrenic nerve. Reflexes. There are 3 different reflexes we had you know. There was the stretch, the Golgi tendon, and the flexor withdrawal reflex. Stretch reflex. What was the sensory input that came into the spinal cord that initiated it? What initiates a stretch reflex? Rapid stretch, thank you. So for the stretch reflex, its stimulus is going to be rapid stretch of muscle. And then therefore its action coming back out is going to be contraction of that muscle. And the whole point of a reflex is that it's actually initiating the contraction of a muscle without getting the go-ahead from the brain. Hey, I want to move this muscle. I would like to contract my quadriceps. It just does it before you even realize it. That's the point of a reflex. It's actually a way for a neurologist to test this pathway. The second reflex is known as the Golgi tendon reflex. What's the trigger for that one? Excessive force on a tendon, we'll just say that. So we know it's a muscle pulling really, really hard, so much that it's about to tear something or break something. So it comes in excessive force. We have an interneuron connecting to this, and the effect in this case is muscle relaxation. The third reflex is known as the flexor reflex, also known as the withdrawal. So it just means take your hand away from something that hurt. So the stimulus is often pain, and the effect is contract, pull arm away, or pull limb away, which often involves contraction of one muscle but relaxing of the opposing muscle. One extra little flip on this is, what if it's a foot? Then it's known as the crossed extensor, because while we're telling our limb, our foot, to pull away, we have to come over here and tell the other leg, brace I need you to contract your muscle so you can hold it up. So you're still doing the withdrawal, pulling the limb away, but if you have it with a lower body, you're also crossing the midline. Again, that's crossed extensor. Cranial nerves. Just know their names. They're Roman numerals. The back of your practical exam, it just has a list. It's just a chart. And there is 12 lines with 3 columns. The first column, just write your Roman numerals. Has to be the Roman numerals 1 through 12. The second column, write them in order. Remember, you want to get your little name recognizers. You have your olfactory, optic, oculomotor, so that's ooh, ooh, ooh. 2, touch, so we have trochlear, trigeminal, abducens, and feel, facial, very, vestibulocochlear, good, glossopharyngeal, velvet, vagus, accessory, and hypoglossal. There are very good Quizlets out there that would have you just do that. And the third column, like something they do, like olfactory, smell, optic, see. I mean, some of them are pretty good gimmies, as long as you got the name down. There'll be some of the multiple choice questions that would refer to the pathology. But on the practical, you don't have to write the pathology out. So if you knew exactly what it did, say oculomotor, eyelid and focusing the lens, then its pathology would be, oh, I have a droopy eye. So you can also just focus on learning what they do. So the couple of questions that do ask about pathology, you'd still be able to know that that's affecting it, rather than a whole nother list.